Hello. Welcome to World of Microbes. In today's video we are gonna talk about the important figures and milestone in diagnostic microbiology. One of the most important discoveries of biology occurred in 1665, with the help of a crude microscope, when Robert Hooke stated that life's smallest structural units were. Antony van Leeuwenhoek is known for his single-lens magnified discovery, with 50x to 300x magnification. Between 1674 to 1723 he wrote series of papers describing his observations of bacteria, algae, protozoa, and fungi named as. Spontaneous generation is the idea that life can come from non-living matter. It was a widely believed scientific theory for centuries, but was eventually disproven. In 1668, Reddy performed an experiment with meat and flies that disproved spontaneous generation for larger organisms. He did an experiment with flies and wide-mouthed jars containing meat. Scientific experiment. Many people say this was the first real experiment. Wide-mouthed jars each containing a piece of meat were subjected to several variations of openness, while all other variables were kept the same. These jars of meat were set out without lids so the meat would be exposed to whatever it might be in the butcher shop. One group of jars were sealed with lids, and another group placed over them. Presence or absence of flies and maggots observed in each jar was recorded. In the control group of jars, flies were seen entering the jars. Later, maggots, then more flies were seen on the meat. In the gauze-covered jars, no flies were seen in the jars, but were observed around and on the gauze, and later a few maggots were seen on the meat. In the sea, maggots or flies were ever seen on the meat. After this experiment, people were willing to acknowledge that larger organisms didn't arise by spontaneous generation, but had to have. In 1670 Jablok conducted an experiment in which he divided a hay infusion that had been boiled into two containers, a heated container that was closed to the air, a heated container that was freely open to the air. Hay infusions are nutrient media to grow ciliates, such as paramecium, which are interesting microscopy specimens. A little bit of dry grass, hay, is boiled with some water. Only the open vessel developed microorganisms. This further helped to disprove abiogenesis. Louis Pasteur disproved spontaneous generation of microbes by preventing dust particles from reaching the sterile broth. In 1861 he completes experiments that lays to rest spontaneous generation. He showed microbes caused fermentation and spoilage. His research in the 1880s demonstrated that thermal processing would inactivate organisms in wine. Spoilage enzymes are also inactivated during pasteurization. Pasteurization is a process in which certain packaged and non-packaged foods, such as milk and fruit juice, are treated with mild heat, usually less than 100 degrees Celsius or 212 degrees Fahrenheit, to eliminate pathogens and extend shelf life. Louis experiment to test whether sterile nutrient broth could spontaneously generate microbial life. To do this, he set up two experiments. In both, Pasteur added nutrient broth to flasks, bent the necks of the flasks into S shapes, and then boiled the broth to kill any existing microbes. He devised a way to flask that allowed oxygen in, but prevented dust. Broth did not show signs of life until he broke off the neck of the flask, allowing dust, and therefore microbes, to enter. Trapped airborne organisms in cotton. He also heated the necks of flasks, drawing them out into long curves, sterilized the media, and left the flasks. The more formal experiments on the relationship between germ and disease were conducted by Louis Pasteur between the year 1860 and 1864. He discovered the pathology of the puerperal fever and the pyogenic vibrio in the blood, and suggested using boric acid to kill these microorganisms before and after. These are some scientists who demonstrations that microorganisms cause disease. Oliver Holmes showed that sepsis could be transmitted by hands of medical student and may cause disease. Louis Pasteur, 1822 to 1895, showed that the pebrine disease of silkworms was caused by a protozoan parasite. He developed other vaccines in cholera, anthrax, and rabies. M.J. Barclay demonstrated that the great potato blight of Ireland was caused by a fungus. Edward Jenner developed the first vaccine and used a vaccination procedure to protect individuals from smallpox. Robert Koch established the relationship between Bacillus anthracis and anthrax. His criteria became known as Koch's postulates and are still used to establish the link between a particular microorganism and a particular Koch's postulate states that the causative, etiological, agent must be present in all affected organisms but absent in healthy individuals. The agent must be capable of being isolated and cultured in pure form. When the cultured agent is introduced to a healthy organism, the same disease must occur. The same causative agent must be isolated again from the development of culture media was important to enable the isolation of pure cultures, only one type of organism. Gelatin was not useful as solidifying agent since it melts at greater than 28 degrees Celsius and some bacteria hydrolyze it with enzymes. Fanny Hesse, the wife of one of Koch's assistants, proposed using agar, since it is not digested and melts at 100 degrees Celsius. Richard Petrie, another of Koch's assistants, developed the Petrie development of vaccines and antisera takes place when Edward Jenner in 1796 discovered that cowpox, vaccinia, induced protection against human smallpox. He called the procedure vaccination. Vaccination is an inoculation of healthy individuals with weakened, or attenuated, forms of microorganisms that would otherwise cause disease detection or active immunity from disease upon later exposure. Pasteur and Roux reported that incubating cultures longer than normal in the lab resulted in attenuated bacteria that could no longer cause disease. Working with chicken cholera, caused by Pasteurella multocida, they noticed that animals injected with attenuated cultures were resistant to the disease. Pasteur and Ch vaccines, attenuated anthrax vaccine, through chemical and heat treatment that is potassium bichromate. Attenuated rabies vaccine, propagated the virus in rabbit following injection of infected brain and spinal cord extracts. Emil von Behring and Shibasaburo Kitasato. Antibodies raised to inactivated diphtheria toxin by injection to different, with the toxin, a toxoid form, antiserum recovered, which contains antibodies specific for the toxin. Protection from disease when injected to non-immune. In 1876 John Tyndall discovered that there were two different types of bacteria, heat-sensitive or heat-labile forms, that is vegetative cells, easily destroyed by boiling, heat-resistant types known as an endospore. Tyndall demonstrated that alternate process of heating and cooling, if repeated 5 times, can kill all the endospores. This is known as sterilization process. In 1876, a German botanist, Ferdinand Kohn, also discovered heat-resistant forms of bacteria. These bacteria are now termed endospores, Bacillus species and Clostridium. The period from 1860 to 1900 is often named the Golden Age of microbiology. During this period, rapid advances, spearheaded by Louis Pasteur and Robert Koch, led to the establishment of microbiology as a science. In 1864 Pasteur established the relationship between microbes and disease in preventing wine from spoiling by using pasteurization. This process kills bacteria in the alcohol by heat, thus preventing the formation of acetic acid, vinegar. His discovery of pasteurization led Pasteur to state that diseases are caused by the growth of microbes in the body and not by sins, bad character, or poverty, etc. Joseph Lister developed a system of surgery designed to prevent organisms from entering wounds. Phenol aka carbolic acid, sprayed in air around surgical incision, decreased number of post-operative infections in patients. His published findings in 1867 transformed the practice of surgery. In the 1890s Ehrlich proposed a theory of immunity in which antibodies were responsible for immunity, antitoxin, known as the father of modern chemotherapy. He speculated about some magic bullet that would selectively find and destroy pathogens but not harm the host, selective toxicity. He also developed a staining procedure to identify tubercle bacilli. Raymond Saboureau developed culture media to study yeast and molds. In 1928 Fleming observed Bacterium Staphylococcus aureus was inhibited in the areas surrounding the colony of a mold that had contaminated a petri plate. The mold was identified as Penicillium notatum, and its active compound was named penicillin. Dmitry Ivanovsky said that tobacco mosaic virus could pass through filters used to remove bacteria. Selman Waxman discovered a number of antibiotics and streptomycin. Resker, 1938, make first electron microscope. The electron microscope is capable of magnifying biological specimens up to 1 million times. These computer-enhanced images of 1, smallpox, 2, herpes simplex, and 3, mumps are magnified, respectively, 150,000, 50,000 and 90,000 times. In 1953 Watson and Crick determined the structure of DNA. They used their research, together with the research of Franklin and Wilkins, to determine the structure of the DNA.